Hello everyone, welcome back to my YouTube channel. My name is Adil. I am a lab technician. In previous video, we have learned about the practical examination of HIV rapid test by TRI-DOT method. In this video, we will learn about the theory behind this practical. Let's start the class. First is aim to detect the presence of HIV in given serum sample. Next is requirements. We need requirements like HIV TRI-DOT kit, micropipette of 50 microliters, and next is sterile tip for the serum transfer. And third is reagents. We have only two reagents. First is HIV buffer, second is HIV conjugate. Fourth is method, that is TRI-DOT method. Reaction, antigen antibody reaction. Next is principle. The principle is based on the immunochromatography by flow assay. The serum is added on the well of the kit. If the HIV antibody present in the serum, it binds to the specific antigen and appears the dot on the test region. Next is procedure. Take the valid HIV kit, place it on the sterile surface. After, first wear the gloves before preparing to the procedure. Add 3 drops of HIV buffer on the well of the kit. Wait for 2 minutes. After 2 minutes, add 50 microliters of serum of the patient and incubate for 2 minutes. After that, again add 3 drops of HIV buffer and incubate for again 2 minutes. After that, add 2 drops of HIV conjugate and incubate for 1 minute. After 1 minute, again add 3 drops of HIV buffer and leave it for 2 minutes. After 2 minutes, you will see the one dot if negative of HIV test. If you see the two dots appears at the HIV kit, the test is positive. Next is interpretation, means the results of the test. The one dot appears on controls means the negative test and valid kit. Next is no dot appears on kit means the invalid kit. Two dots appear, one at HIV1 and at one at control is known as HIV1 positive. The two dots appears at one at HIV2 and control positive for HIV2. Three dots appear at HIV1, second and HIV2 dot, and third at control. HIV1 and HIV2 both positive. Hope this video is helpful to understanding the theory class of HIV rapid test by TRI-DOT method. Please like, share and subscribe to my channel ADILlabtech 555. See you in the next video.